In this video, I'm going to continue talking about chemistry, but I'm going to specifically talk about biochemistry. So biochemistry is the study of compounds that make up life. All biochemicals are organic compounds. They're typically covalently bonded, like we talked about before, so they're carbon-based as well as what an organic compound is. So there are four major macromolecules that make up the cell. They are carbohydrates, lipids, proteins, and nucleic acids. And we're going to go into detail about them as well as talk about some other important biological compounds. So carbohydrates are sugars, also known as polysaccharides. And remember, in the last lecture video, I showed you those rings. So remember, those rings represent rings of carbon. So when you have one carbon ring, it's a monosaccharide. When you have two together, bonded together, it's a disaccharide. And when you have a big long chain of them, it's a polysaccharide. And so some glucose is a very important carbohydrate. And there's some other important carbohydrates that are specific to microorganisms and bacteria that I'll talk about here in a second. So they exist as monosaccharides with 3 to 7 carbon rings, disaccharides with 2 monosaccharides together, or polysaccharides where you have a big long chain of these sugars. The functions of a polysaccharide in the cell is to create structural support and protection, so they make up cell walls. They serve as nutrients and storage, so glucose is a nutrient molecule. We can also create polysaccharides or sugars within the cell that can store energy as well. Cell walls in plants and in bacteria are made up of polysaccharides. In the case of like algae plants, the polysaccharide is called cellulose, and you can see that they actually make, instead of being one long chain, they make these chains that then have these cross bridges, and they're very strong. Other important polysaccharides that we'll learn about in microbiology is agar. Agar is an important solidifying agent for media, so it's what makes bacterial media gelatinous, and we saw that in the lab. Peptidoglycan. Peptidoglycan is extremely important. It makes up part of the bacterial cell wall, and it is only found in bacteria, which makes it specific to bacteria. Chitin is found in shells. Glycocalyx is actually what makes like a slime layer or a capsule coating around bacteria, and next lecture I'll go into that more in detail too. So there's other important saccharides that are, or polysaccharides that are energy storage and building that are specific to microbes that we'll talk about throughout the semester. The next type of macromolecule is a lipid. So lipid's a fat or a wax or a phospholipid, and they're not soluble in polar substances, so they're non-polar. So triglycerides are an example. Fatty acids, fats, oils are all examples of lipids. But the most important lipid is the phospholipid, and the phospholipid contains 2 fatty acid tails, and it has a phosphate head. So it's got this is the phosphate group. It's a phosphorus with 3 oxygens, 4 oxygens, and then you have the fatty tail. So the fatty tail is actually nonpolar, and the phosphate head is polar, and that's what creates the phospholipid bilayer, and it prevents water from going in and out of the cell. So inside you have these 2 layers of phospholipids, and they create sort of this waxy, fatty interior where water does not go, and then you've got these charged polar heads that attracts water outside and within the cell. Some other lipids are steroids or cholesterols, and these are important in stabilizing the cell wall. They're found embedded with, the cell membrane. They're found embedded within the cell membrane and help to stabilize it somewhat. So you can see they're like, here's the phospholipids, and all these little yellow things are cholesterol. And so it just kind of like holds it together. Proteins are the third type of macromolecule, and they are made up of amino acids. There are 20 different amino acids that come together, and they form peptide bonds when they come together, and then they form these big long chains that then develop into a protein. And proteins are essentially what allow the cell to function and undergo like chemical processes. So as we said, we've got these big long chains of amino acids, right? So that is the primary structure of a protein, where you have amino acids. Each one is represented by this little blue circle here, and they come together in a chain. That's the primary structure. Then what happens is the chain starts to wrap around itself in either alpha helices or beta pleated sheets. And this is the secondary structure. So parts of the chain are attracted to one another when it folds over itself. Then these big helices or pleated sheets start to fold in on each other even more, and there's these strong, what they're called disulfide bridges or disulfide bonds that give it its three-dimensional or tertiary structure. A functional protein, however, is typically what is thought as a quaternary structure protein, where you've got multiple tertiary structures that come together in a certain way in order to produce a full structural protein. So if you look at here, this is actually a diagram of a protein, and each different color is supposed to represent a tertiary structure. So you've got red and yellow and blue and green here, and you can see the alpha helices and beta pleated sheets of the secondary structure, and they all come together in a very specific shape, which then forms the actual functional protein or enzyme. Protein shape is unique per protein, and it's important for what it does. Examples of proteins are enzymes and antibodies, which are very important for any sort of chemical function or metabolic function within the cell. So the native state of a protein is the three-dimensional shape in which it is intended. A denatured protein is when its native state is disrupted somehow, and that can be done by changing the pH or heating it up, and essentially it makes the protein non-functional. All right, the fourth type of macromolecule is a nucleic acid. And nucleic acids, there are two forms. There's DNA and RNA. And they are made up of units called nucleotides. And the nucleotides have three different parts. They have a nitrogen base, a sugar, a pentose sugar, and a phosphate. So the nitrogen base can be either purines or pyrimidines. The types of purines are adenine and guanine, and they're double ringed, so you can see them here. See these rings? If anything that's not marked as a carbon, so it's rings of carbon and nitrogen. And then you have a pyrimidine, which is one ring, and there's three different kinds. There's thymine and cytosine and uracil. Thymine and cytosine are found in DNA only, or in DNA. Uracil is found in RNA, and that's a substitute. Instead of thymine, RNA has uracil. The type of sugar that is bonded, it's a ribose sugar in RNA, it's a deoxyribose in DNA. And then, of course, they have a phosphate head, just like the phosphate of the phospholipid. It's a phosphorus with 4 oxygens. Okay, so they come together. So here you can see this is a nucleotide. It has a nitrogen base, which again, remember, is the purine pyrimidine. It's the adenine, guanine, cytosine, uracil, or thymine. And then you have the sugar, and then you have the phosphate, and they come together in these chains. So here's your nitrogen base, your sugar, your phosphate. Then this phosphate attaches to the sugar here, and you have your nitrogen base, okay? And so it creates this backbone right here. And this backbone, these strong bonds right here, are called phosphodiester bonds. They're very strong bonds. Now, DNA is anti-parallel, so essentially you have two different strands that come together, and the nitrogen bases of adjacent pairing, complementary base pair, bond by a hydrogen bond here, which is a fairly weak bond, which is why it's shown by dashes, so that it can be split open. So the double helix is what forms when they come together and they wrap around one another, and the base pair is complementary base pair, like I said. So adenine goes with thymine, cytosine goes with guanine. In the case of RNA, instead of thymine here, we would have uracil. So RNA is a type of nucleic acid, and RNA is important in terms of protein synthesis, but also some other functions within the cell. And there's several different kinds of RNA. So DNA really codes for proteins and is sort of the message holder, the code of life, whereas RNA helps to express the DNA. So it's a long chain of nucleotides, but it's, we already discussed that one difference, that they have uracil instead of thymine. And there are a couple of different kinds. You have messenger RNA, transfer RNA, and ribosomal RNA, and you can actually see them all right here. And we'll go into detail about genetics as we go on. This should be a review from standard biology, but we will review it again when we talk about genetics. So this is a ribosome here. This is where a protein is made. This ribosome is made up of, there is some RNA in it that makes it up structurally. The messenger right here is what has been created from the DNA template. And these are the transfer RNAs right here that help to create the amino acid sequence. Right? So, again, that should be review, but we'll go into more detail about it when we get to genetics in case it's been a while. Now, there's a couple other molecules other than the 4 macromolecules that I want to talk about, just so we know they're very important biological molecules. The first is ATP. So ATP is the energy storage molecule. It's adenosine triphosphate. So essentially a nucleotide that has adenine, ribose, and it has phosphates at the end. So instead of just one phosphate, you add multiple phosphates. And what happens when you attach these phosphates to one another, so you see one right here, and then another one attaches right here, another one attaches right here. So that's why it's called tri, because there's 3. If it's just 2, it's adenosine diphosphate. If it's 1, it's adenosine monophosphate. And these bonds right here, these little red squiggles, are extremely high energy bonds. They store a huge amount of energy. So the cell works to build these bonds by, taking energy from the external environment and building these storage bonds, and then holds on to them until they can be broken and the energy from that can be released to do something else in the cell, like build a phospholipid or build a protein. Okay, so ATP is the energy storage molecule of the cell. So those are the important large biological compounds that we need to have reviewed in order to move forward, and now to start talking about cell structure.